National Advisory Eye Council, 6700B Rockledge Drive Conference Center, NEI, NIH, October 12, 2017. Retinal Neuroscience Portfolio Overview, Thomas Greenwell, Ph.D., Program Director, Division of Extramural Science Programs, NEI.
Thank you for giving me the opportunity to talk today about my portfolio. It is a very broad portfolio.
Slide, Retinal Neuroscience Portfolio Overview, Thomas Greenwell, Ph.D., Program Director, Division of Extramural Science Programs, NEI. Image of retina bipolar cell types.
I have a lot of material, but I'm going to try to give you about a 5,000 to 10,000 foot view of some of the highlights in the portfolio. Won't be able to talk about everything, of course. This is just a nice picture of different bipolar cell types in the retina. And as Dr. Steinmetz mentioned, I have the retinal neuroscience portion of the portfolio. I'll get into some of the definition of what that portfolio is, and again, some of the recent advances, and we should have some time for questions at the end of the talk.
Slide, NEI Funding by Program. Pie chart, order from the largest is retinal diseases, 44%, SAVP, 23%, corneal diseases, 13%, glaucoma, 10%, lens and cataract, 7%, and low vision and blindness rehabilitation at 3%.
So you've all probably seen this before. This is the pie chart of how funding programs work in Extramural National Eye Institute. And the retina portfolio makes up 44% of the funded grant applications. And this number comes out of success rate. We keep the success rate even through all of the programs. So if 100 grants come in for retina, and we're able to fund 20% of them, then or 44% of them, then there would be 44. And the same thing is true. We keep that funding percent the same for the other programs. So it's really a denominator effect here. My program is a little less than a third of this retina composition.
Slide, the structure of the retina. Image of the retina structure showing various components and divided into 7 layers. From top is the RPE, photoreceptor outer segment, POS, outer nuclear layer, ONL, outer plexiform layer, OPL, internuclear layer, INL, inner plexiform layer, IPL, and the ganglion cell layer, GCL. The bottom 5 layers are circled. From Santos Carvalho et al., 2013.
So what might my program be? Well, first we'll talk a little bit about the retina, and I'll get to the circle in a moment. But as you know, in the retina, the light comes through the vitreal side, through all of these cells, and then hits the photoreceptors, and then the photoreceptors transduce that light, the photons, into an electrical signal. And that crosses the synapse and bipolar cells and has processing through the horizontal cells, bipolar cells, amacrine cells, and the retinal ganglion cells until finally they pass through the nerve fiber layer to the rest of the brain. There's also some other support cells in here, and the reason this is circled is that the development and the physiology of most of these cells in the circle is a big part of the portfolio.
Slide, composition of retinal neuroscience portfolio, FY 2017.
As of the fiscal year that just finished, I have 140 grants in my portfolio. Some of those are held by more than one PI, as you may assume. 39% of the grants are development of the retina, and this will give you a little peek into the organization of my talk. I'm going to talk about cell lineage and connectivity and synapse formation briefly, about retina development. And then primarily I will be talking about retinal circuitry and signal processing.
66%.
Talking about cell typing, the parts list, and also how those cells connect to each other. And I'm going to use the direction selective circuit as an example of connectivity and signal processing. And then I'll also be briefly touching on retinal regeneration, which is 19% of my portfolio.
Regeneration of retinal ganglion cells. Regeneration of amacrine and bipolar cells from Mueller glia.
And technology development.
16%.
Which is has to do with a broad part of the portfolio, but I'll talk about 3 examples: prosthetics, optogenetics, and imaging.
Slide. Areas of major advances of the past approximately 10 years.
Some of the major advances of the past 10 to 15 years has been cell typing and connectomics, direction selective circuits in the retina, the use of computational neuroscience in the retina, and from that determining population function. One of the big tools for this finding was the multi-electrode array, which I'll talk about, and also retinal regeneration and reconnection. I will note that there are other advances. I just don't have time to talk about those. One notable advance that I'm not going to be able to talk about is the melanopsin cells, determining that those retina ganglion cells have light sensitivity.
Slide. Development of the retina. Numerous groups have determined factors needed for birth of different retinal cells. Left image shows cell lineages in the retina. Source: Bassett and Wallace, 2012. Right image shows how lineages derive from retinal progenitors. Source: NEI et al., 2017.
So briefly, development of the retina. Again, here's another schematic of the thin layer in the back of the eye. And we know from this review in 2012 that the cells are born at different times from embryonic day 10 for retinal progenitor cells at the bottom here, all the way to postnatal day 12. And there are numerous factors, which I'm not going to get into, I don't want to put anyone to sleep right now, that are involved in making these cells. And the timing and mixture of the factors are pretty well worked out, although there is ongoing research on these studies. But this helps build the parts of the retina. And not only do we know some of the overall subtypes, but this picture over here gives an example of how subtypes of amacrine cells are known to form with certain transcription factors. So if you go down this pathway from a retinal progenitor cell with NeuroD1, NeuroD4, and then it can split into this amacrine precursor, can split into either a glycinergic amacrine cell, a GABAergic amacrine cell, or some other type of amacrine cell. Showing a gap in knowledge is that they are not quite sure what transcription factors might be involved as of 2007 for what amacrine cells make.
Slide. Development connectivity. Bar charts for ELFN1 and mGlu-R6 showing specific reduction in ELFN1 and mGlu-R6 in alpha-2 delta-4 subnet knockouts. Source: Wang, Marty-Mayanov, Neuron, 2017. Image illustrating CAV 1.4 ELFN1 and the alpha-2 delta-4 subnet on rods connecting with mGlu-R6 on an on bipolar cell across a synapse. Source: A Line in a Synapse, Kirschensteiner, 2017.
Now to connectivity. While we know how the cells are made and what mixture is needed, but how do synapses form? How does synapses form in the nervous system? This is a recent finding by an example of how synapses form from the Martin-Mayanov lab at Scripps, Florida. And it was determined that a calcium subunit, this alpha-2 delta-4, is involved in actually helping the complex from a rod synapse form to an on-rod bipolar cell. And this also helps with the cones in the arrangement with connexin 36. This was previewed in that issue of Neuron by Dr. Kirschensteiner, and this schematic gives you an idea of also the special synapse that rod cells have along with rod bipolar cells and how this special synapse is formed. This is giving us a lot of insight into not only how the nervous system in the retina connects, but potentially if we want to rebuild those connections. So this is a very high impact finding, and we expect a lot of our other researchers to give us more insight into synapse formation in the retina.
Slide. Types of cells in the mouse retina. Image showing types of cells.
This is just a depiction anatomically of the different cell types that were known in 2001, and a very nice paper by Richard Masland in 2001 We knew there were 3 different types of photoreceptors. That has not changed much. 2 horizontal cell types, 10 bipolar cell types, 24 amacrine cell types, and 10 retinal ganglion cell types. And we now know that this estimation is actually a lowball number based on some of the recent findings.
Slide. Types of ganglion cells in the retina, approximately 2016. Image shows functional RGC types of the mouse retina. Source, T. Baden et al., Nature 1-6, 2016. DOI:10.1038/Nature 16468.
So for now, functionally, a paper that came out in just last year was determined that there's about 40 different types of retinal ganglion cells, and that's broken down by this very small graph with a lot of information. After looking at anatomy and physiology and connectivity, it's probably closer to about 30, but as you can see,
Return to slide. Types of cells in the mouse retina.
a jump from 10 in 2001 to 2016 being 30 is a pretty big jump. The same is true for bipolar cells as well.
Slide. Cell typing. Studies using DropSeq. From Sains, Nature Reviews, Neuroscience 2017. Mouse retina, 44,808 cells sequences. Mouse retina bipolar cells, approximately 25,000 cells sequenced. Various cell typing images.
And again, I've just reviewed here some of the cell typing. So there's genetic techniques, very recent, a new finding by Josh Sains' group, along with Hongqi Zheng at the Allen Brain Institute, funded in part by the Brain Initiative here at NIH and across agencies, found that there were 39 overall cell types in the retina and 15 bipolar cell types using this high-throughput DropSeq technique. When you combine, again, anatomy, electron microscopy, confocal connectomics, and physiology, then you get a good picture. And we actually have just funded a new investigator, Bart Borghaus, who's going to functionally type bipolar cells as well. So I'm not trying to show this as end-all, be-all, but work is ongoing on cell typing as well in the portfolio for a more comprehensive view.
Slide. Connectomics. Images of jumbled wiring. Connectomic reconstruction of bipolar cells and starburst amacrine cells by the iWire community. Source, Sung et al. And A2 amacrine cell circuit by the Mark Lab, Utah. Source, Mark et al. 2014.
You may have heard several councils ago, Sebastian Sung came and talked about connectomics. So what is connectomics? Well, we know a lot of the cell types, but we don't know the messy wiring. And this is just a depiction of how the wiring can be quite jumbled, not only in real life, but possibly in the nervous system. So how do we untangle and decipher that wiring? And the best way that we have now is high-throughput electron microscopy that groups like Sebastian Sung are doing using iWire, which is a community video game, to solve these connections. There's another group, and this is actually an example of the direction selective circuit, bipolar cells and starburst amacrine cells. This is another picture from the Sung group. I also want to highlight another group working on the A2 amacrine cell circuit in Utah. And they are, again, this is a very beautiful picture that shows the cell types and the connections that the amacrine cells will make to bipolar cells and other cells, and sort of how they're tiled across the retina as well. So there is quite a bit of organization in the retina that we've known for many, many years, the days back to Ramon y Cajal, but the specific connections and the circuits, microcircuits, we're working out.
Slide. Direction selectivity in the retina. Direction selective cells theorized to present in the retina. Barlow and Levick, 1965. Cells responded with preferences for 4 cardinal directions. Image shows preferred and null directions for light entering the cell. DS cells were not chemically identified until 1990s/2000s, with synaptic mechanisms identified in early to mid-2000s. Taylor, Werblin, Zhao, etc. Importance of the starburst amacrine cell. Yoshida, Neuron, 2001.
So I want to talk about direction selectivity, because this is an issue that started back in the 1960s by Barlow and Levick. when they first decided that from data that there were specific cells in the retina for direction selectivity. And why am I using direction selectivity as an example? Well, it's one of the ways that the retina processes a signal. All of the light and other visual information that comes into the eye and hits the retina, luminance, contrast, motion, which I'm going to talk about here, direction selectivity, color, that all goes through signal processing before going on to the rest of the brain and the cerebral cortex, V1, and so on. So again, this is being worked out. If we don't know what the rest of the brain is getting, what V1's getting, then it's really hard to look at vision in that process. So again, direction selectivity was theorized to be occurring in the retina in the 60s, but that wasn't worked out until very recently. And one of the big discoveries was in 2001 with the importance of the starburst amacrine cell. This is from a paper in 2012 where it was determined that some of the mechanisms that Barlow theorized are happening in the retina and can be physiologically described, where this cell has a preferred direction to the right, and by moving a bar either to the right or to the left, you can record the responses from A and B as well as from the cell. And you can subtract those responses, and that's what the cell is doing to give these on and off signals in the preferred direction versus no signals in the null direction.
Slide, direction selectivity in the retina. Images of starburst amacrine cells from A. Borst and T. Euler, 2011, Kirschensteiner, 2015, and Zhao, 2010.
So what are the mechanisms of that? Well, Borst and Euler have a nice review on that, where starburst amacrine cells that are both cholinergic, excitatory, and GABAergic, inhibitory neurotransmitter. And that was a big discovery in 2010 by Jimmy Zhao, showing that these starburst amacrine cells have both acetylcholine and GABA in them and have physiological responses to agonists being put on. So if the preferred direction of this cell is to the right, part of the explanation, again, is timing and response of the bipolar cells and the amacrine cells here, but also the direction selective cell gets selectively inhibited by the direction and the GABA receptors that are on this synapse. So again, a bar moving to the right would activate the cell very strongly. A bar moving to the left would not, because it could activate the cell to release more inhibition at this synapse. And again, this circuit is still being worked out. In fact, a new cell was discovered by the Kirschensteiner lab in 2015. And it's actually a very complicated circuit. There's about 6 or 7 different types of direction selective ganglion cells. So there was a new type of amacrine cell, a VG3 glutamatergic glycinergic cell that could be involved with the W3 type of direction selective ganglion cell.
Slide, how the direction selective circuit is built.
Moving on, how is this circuit built? Well, what's interesting is that the circuit is in the retina before eye opening. It's independent of vision. Many developmental biologists know that retinal waves are involved in organizing the retina, and it was theorized earlier that retinal waves might be involved in organizing this direction selective circuit, but they're not. Blocking these waves does not stop the circuit from forming. So it's independent of inhibition of the retinal waves. And a very nice review from Feller
points to image on right, divided into 4 sections labeled A through D, from Mori and Feller, 2016.
Has shown that there's actually an asymmetric development, asymmetric inhibition built into the circuit.
Points to section C within, which shows GABA receptors and the preferred direction from right to left.
Where these are the GABA receptors down here on your lower right, and there's less GABA receptors on the preferred side. So if the direction is to the left, I know I'm reversing right and left on you, but if the direction is to the left, then there's going to be more GABAergic inhibition on the left. We're also working out, just this picture on the left,
Image from Duan Sains, 2014.
shows a lot of the molecules and signaling and timing of how the circuit forms. We know that cadherins are very important for the direction selective circuit and the wiring up for the type 2 and type 5 bipolar cells, also for the amacrine cells and the ganglion cells that are involved in the formation of the circuit. This picture here
Points to B and D in the picture on the right.
just describes the different types of cells and different directions in which they're responding to. So these are 4 different types of retinal ganglion cells responding in each of the 4 directions.
Slide, signal processing in the retina. Images representing multi-electrode array and a schematic showing processing from a visual environment to photoreceptors, then ganglion cells on the retina to code, leading to behavior.
Next, I'm going to move to signal processing. I'm still going to try to use direction selectivity as an example of this. But again, a big finding is the multi-electrode array. These electrodes were engineered to have 512 electrodes on them. So you can actually place a retinal slice onto this electrode. And this gives you an idea. The retinal slice is still alive and still responding to visual scenes. So you could surmise that a visual scene of a cat would activate the retinal slice, the ganglion cells in a certain way. Again, there's different types of ganglion cells. And that the animal eventually would have a outgoing behavior from the neural code translated in these retinal ganglion cells. So I want you to remember that each cell is giving a different code to the rest of the visual system, because I'll get back to that later.
Slide, information processing in the retina. Left image shows different directional ganglion cell responses from multi-electrode array recordings. Source, Nathan Schwartz, J. Neurosci, 2016. Right image shows cones connected to on and off midget cells. Source, Field et al., 2010.
So information processing. So on the left here is work from another new investigator in our portfolio, Greg Schwartz at Northwestern, and he's discovered using computational methods and multi-electrode array recording that different types of direction selective ganglion cells that respond in different directions actually correlates to their dendrite outgrowth. So that's one example of computational neuroscience electrode array, discovering some new things about the direction selective circuit. I want to talk a little bit more about the multi-electrode array in a significant finding from 2010, where they were actually able, this is Greg Field and E.J. Chichelnitsky, were able to record from the primate retina slice on a multi-electrode array and determine the cones that actually connected to each individual retinal ganglion cell. So this ganglion cell here has a majority of green cones connected to it, as well as red and some blue. This one here has a lot more green cones connected to it, more blue, and very few red cones. And you can see that in this depiction here. And some more computational work was done here, looking at the relative distribution in the retina of cone connectivity for blue, red, and green. I'm going to switch gears to retinal regeneration. This is an overview slide. There are quite a few groups in the portfolio.
Slide, retinal regeneration, retinal ganglion cells. Image, reprogrammed Mueller-Glia cell regeneration cycle. Source, Goldman, 2014.
Trying to push Mueller-Glia cell regeneration. So the cells, if they can push them to progenitor cells in the retina, and then those progenitor cells can make cells that you would want to fix the retina, such as photoreceptors or retinal ganglion cells or the other cell types. In my portfolio, I have, again, retinal ganglion cell regeneration, amacrine cell, and bipolar cells. You will hear from Dr. Newhold in January about photoreceptor regeneration, since her portfolio is comprised of photoreceptors and the RPE, retinal pigment epithelium. So this is a specific example of pushing Mueller glia cells to make progenitors for retinal ganglion cells. And this is easily done in fish, but the problem, and you might have heard about from Dr. Sieving this morning, the problem is really pushing this into higher order mammals. And that's a good part of the Audacious Goals initiative, and some of this is work that we're already funding in the portfolio before the Audacious Goals initiative started. And we know that numerous factors can inhibit or stimulate regeneration, and that by leveraging this, we might be able to push regeneration in mammals and eventually get therapies. So a lot of the portfolio is understanding this Mueller glia cycle, the push towards regeneration, and what kind of cells might come out of that.
Slide, retinal regeneration. Example, regeneration and reconnection of amacrine and bipolar cells. Right image shows reconnection of regenerated cells in the retina with GFP marker. Jorstad et al., 2017. Lower left image shows physiological functional responses to light.
And a specific example and finding from just this year by the Tom Ray lab is that he found two factors that worked in the fish. One is ASCL1, that's the name of it. And if he pushes ASCL1, which with a histone deacetylase inhibitor, that actually pushes regeneration in a mouse. And not only regeneration, but reconnection. So this panel on the right shows reconnection of these regenerated cells into the retina with GFP marker. And the panel on the lower left not only shows the reconnection, but actual physiological functional response to light. So this is a big finding. He's been able to do this with amacrine and bipolar cells. I think the large push is to try to do this with a sufficient number of cells and photoreceptors, since most retinal diseases affect photoreceptors rather than amacrine and bipolar cells. There are some diseases that cause amacrine and bipolar cell regeneration, or where those cells are affected, like congenital stationary night blindness. But again, pushing this in a higher number is really important. And this is a nice, significant, comprehensive paper, which we need to look forward towards animals beyond mice.
Slide, technology development, prosthetics. Images of electrode, amplitude, and type stimulus. Source, Weiland, Neural Engineering, IEEE Proceedings, 2017. Image of amplitude and current over distance. Source, Fried, Journal of Neuroengineering, 2011.
Another part of the portfolio is technology development. So I'm shifting gears a little bit from regeneration to prosthetics. We all know that retinal prosthetics has come a long way in the last 15, 20 years. But a lot of those prosthetics patients can't see very high detail. So the issue for preclinical work in my portfolio is trying to understand stimulation methods, and that's just what I'm showing here for technology development, through neural engineering, either engineering better electrodes that can stimulate more specifically, or stimulating methods by changing amplitude or type of stimulation. And this is from an abstract by Jim Weiland in Neural Engineering just this year, or by looking at area in space and amplitude current. And this is by Shelley Fried in Neuroengineering. So this is not an old idea that you could potentially stimulate dendrites or axons with different amplitudes and currents. Then you might be able to stimulate a cell body. But trying to tweak this physiology and stimulate in a more specific type so that the resolution of already existing electrodes can be improved. So that's part of the work in my portfolio.
Slide, technology development, optogenetics. Image of blue light-sensitive CHR2 sodium channel tagged with RFP. Image of channel rhodopsins in bipolar cells. Source, PAN.
Not only are prosthetics being used, but there are also molecular approaches to restoring vision. And a lot of that, when it hits prime time, is in Dr. Shen's portfolio, but before it hits prime time, sometimes it's in mine. I know Dr. Shen gets a lot of the exciting stuff, but I have some exciting stuff too. So if you don't know about optogenetics or channel rhodopsins, well, they were discovered in bacteria that lived in lakes in the Middle East, and these bacteria had eye spots so that they could go to areas of higher light, so that they could live, make fuel. So very smart researchers were able to engineer these channels that induce excitation, and this is just an example of a channel that also emits red fluorescent protein when green light excites that protein, and blue light excites the channel, gives sodium, and if you're not a neuroscientist, sodium is the main activator of the nervous system. So there are a lot of groups working on optogenetics. Dr. Pan, this is a picture from his publication several years ago where he's able to put channel rhodopsins into bipolar cells. So the thinking is that if photoreceptors are lost, you can confer a response to light in the next cells down in the retina, the bipolar cells. And one exciting finding from last year is that Dr. Pan has been able to express channel rhodopsins in a higher mammal, the marmoset. And that is what these pictures here.
Image, marmoset retina. Source, Liu et al., Gene Therapy, 2016.
Using two different vectors are showing is that this cell in red are those that have the channel rhodopsins and responsive to light. And the other exciting thing about this project is that it's moving into clinical trials via a company that was formed a few years ago called Retrosense. And so there is some hope for restoring vision using optogenetics. I will talk about optogenetics a little bit briefly later on when I get to combinatorial or translational approaches in the portfolio.
Slide, Technology Development, Imaging-Adaptive Optics Scanning Laser Ophthalmoscopy. Left image, complex arrangement of mirrors and detectors.
The next piece of technology development I want to give a brief overview of is adaptive optics scanning laser ophthalmoscopy. I have to say it slowly or else my tongue twists. So the thinking behind adaptive optics, which was first developed in astronomy many years ago, but has been adopted in vision sciences not too long ago, is that you can correct for aberrations in the sky for astronomy, but in the cornea lens, vitreous, in the visual system, in vision science. And so that's what all these complicated mirrors and computers do. Again, I'm not going to get in great detail. I've seen this in Austin Rorter's lab and others' labs, and it's quite a setup on a table. It almost looks a little bit like my desk, only not as messy as my desk. But this example...
Right image, correcting for eye motion, showing zoomed high-resolution segment. Source, Ruruda, 2015.
Is correcting for eye motion. The other thinking behind using this approach is better resolution of cells in the retina, in the living retina in a human. So not only could you see cones, which is what this picture on the right is, but potentially researchers are trying to find out how to see retinal ganglion cells. And a lot of this is important for disease progression, but also monitoring how therapies might eventually work, especially if we're doing cell replacement therapies. And that was one of the first initiatives funded by the Audacious Goals Initiative. We have several groups working on improving imaging in the retina for this purpose. And this is a nice advance from 2015, again, correcting for eye motion. Many people who have visual disorders have nystagmus, where there's a tremendous amount of motion in the eye, and they're getting very close to being able to visualize these rods and cones in very high resolution, even with nystagmus.
Slide, bringing it together, translational work, firing patterns of the retina's output cells. Contains image and pattern data while viewing movies. The The resolution from the blind retina through the standard prosthetic is poorer than normal vision. Encoder CHR2 prosthetic restores near-normal encoding. Source, Nierenberg.
So how does this all come together? I want to show, again, just one example of how different approaches and different advances can come together by looking at preclinical and translational work in the visual system. And this is work by Sheila Nierenberg at Cornell, where she is actually a mathematician, and so is E.J. Chichelniski, and very smart people who Dr. Nierenberg has claimed that she knows the language of the retina. She knows what the output of the retina is to a corresponding visual scene. And thus, if the photoreceptors, bipolar cells are lost, she can actually understand the code for those retinal ganglion cells. So that's what this picture is here on the top. It's firing patterns in 5 cells during viewing a movie. And I'm going to skip down to the bottom. A blind retina viewing movies would have sort of a jumbled response similar to what you see on the top, but again, probably not very high resolution. So the thinking is, again, improving resolution for people who have been blinded and when they get a therapy. So Dr. Nierenberg has been able to use this encoder that encodes responses from a visual scene for specific cells. And she can use a channel rhodopsin prosthetic, which I'll explain on the next slide in a little more detail, to basically mimic very closely what a normal retina would be seeing. And you can see that here visually. Here's the original image. Here's the encoder itself viewing the image. So pretty good resolution to the original image. And then the blind retina viewing the image through the encoder and channel rhodopsin prosthetic versus a blind retina viewing the image through just a standard prosthetic. Again, no face recognition. Many clinicians will tell you that retinal prosthetics give a person the ability to see light versus dark, sort laundry, but not see faces. And so the hope here is that at some point in the very near future, people can have face recognition who've been blinded.
Slide, translational work. Image illustrating how an encoder could assist brain processing an image.
So how would this work? Well, again, I've sort of explained the encoder would sort of replace the processing in the retina, since those cells would be damaged, destroyed, or traumatized. And if you basically put this computer chip or encoder into an eyepiece that not only can take light in, might be able to amplify it, because we know that channel rhodopsins have a very low level of light response. You need a very high light intensity. And if they can amplify that intensity and then use the encoder to tell the rest of the visual system what the retinal ganglion cells would be translating from that visual scene, then you are essentially able to replace that retinal processing with the chip. So that's the hope.
Slide, in summary.
Still lots of work to be done. Again, I've briefly talked to you, hopefully not too quickly, about the development of the retina, cell lineage, the connectivity and synapse formation. We're understanding the molecules and how the cells are built in the retina, how the signals process through the retina, through the retinal circuitry, that we've known the basic circuitry for many, many years, but the circuitry is being resolved at a microcircuit level through connectomics and other approaches. Through cell typing, we're getting a full parts list through three different types of approaches: genetic, anatomic, and functional physiological. Understanding that information processing, the language of the retina, and then, again, regeneration, very important part. If we can regenerate those cells in the nervous system that have died or have been damaged, and we're getting there with retinal ganglion cells and with endocrine and bipolar cells, as you saw from Dr. Ray's work. And then technology development, improving resolution of prosthetics, optogenetics, and molecular engineering, as well as imaging. And I hope I gave you a good translational example. It's exciting. Dr. Nierenberg is trying to push this into humans and get funding for that. She's won a MacArthur Genius Award. She has a TED Talk. If you don't understand what I said, you can look up her TED Talk. She probably explains it better than me. But it is exciting to be able to see this broad portfolio. I feel very privileged.
Slide. Thanks. Cartoon illustrating input, then a miracle occurs, then output with a quote. I think you should be more explicit here in step 2.
And hopefully today I explained a little bit about the middle part, where light comes in, gets translated by the retina, and then there's some sort of output. And we know more than a miracle occurs in the translation of a retina, that there actually are some mechanisms for that. I'd like to thank all the scientists in my portfolio. Again, I feel like an observer on the fringe of amazing findings, and I'm very privileged to be in this position. And I'd also like to thank my coworkers for feedback, for always bouncing ideas off of both them and myself. And thank you for listening to me today.
Logo for NIH, National Eye Institute.